Every once in a while there's a flurry of activity in blood substitutes, and people simply ask whatever happened to them. I would like to disclose that I do have some potential financial conflicts which do not, in fact, affect this talk. On the other hand, I have some strong feelings about groups like BioPure, who I consider a criminal activity, and so does the US government. These are the sorts of people who would walk up to you in scientific meetings and saying, "I'd be careful about what you say. You have a nice family." Or they'd call C. Everett Koop to call your boss and tell him that you're incompetent. This is the way the field went. And so this is the history of publications on blood substitutes over the last 80 years. And it really reached a peak in 1992 with the publication of the Baxter trial, and again in about 2006 with the publication of the Northfield trials, and has sort of fallen off since then. But let me go back and talk about the basic history. The first person to ever put a syringe into a vein was Christopher Wren, the man you know as the architect of all those churches in London. Stuck in Oxford during the English Civil War in Robert Boyle's sister's basement, they played with the syringe on his vacuum pump and injected beer and wine as substitutes for the vital spirit of blood. It says in Robert Boyle's notebook that ye dog did piss mightily. Over the last 200 years, many objects have been used as blood substitutes. With the development of oil immersion lenses, it became clear that blood was corpuscular. And so the fact that milk appeared to be corpuscular as well led to its infusion. Saline solutions were developed in the 1880s to treat cholera. Colloids were recognized as at least part of what was involved in blood because you could show that they would absorb water. And so things like gum acacia and gelatins, polyvinyl pyrrolidone, extending into our own era with dextrans and hetastarch. It's only been recently that we've recognized that these materials do poorly when used as blood substitutes. More recently, perfluorocarbon emulsions were shown to carry oxygen, and hemoglobin solutions can support life in the absence of in acellular forms. During World War II, we made almost 10 million units of pooled freeze-dried human plasma and literally issued it to every medic. And it's simply pooled freeze-dried plasma. The second can contains the distilled water that you reconstitute it with a long rubber tube with a needle on each end and a second needle to let the air out. And this was widely used. Okay. Only two materials, however, carry oxygen. You can dissolve oxygen in perfluorocarbons, and you can bind oxygen with hemoglobin. The first person to actually explore this in a systematic way was George Minot, the man you know as the Nobel laureate for liver feeding in pernicious anemia. He was a diabetic. You can see he's quite thin. He starved himself to avoid ketoacidosis and was kind of interested. His friends got him a job as the research doc at Boston City Hospital. And one of the questions he came up with is, what happens to hemoglobin when cells break down? And so he took red cells and gently added distilled water until they expanded, leaked the hemoglobin, and collapsed. And then he took the hemoglobin solution and added salt back to make it isotonic and injected it into people, and couldn't see that it did anything of serious harm and did not know where it went. A decade and a half later, Amberson at the University of Maryland did the same thing with a large amount of cow blood and exchange transfused a cat to a hematocrit of less than 1. And taking the cat, he held it upside down and dropped it, and it landed on its feet and walked away. And so he said, look, neurologic activity occurs, when oxygen is transported by extracellular hemoglobin. During the war, the Army looked extensively at cell-free hemoglobin and decided that the issues of hypertension around its use and the fact that, you'd give it, people would get hypertensive, and then all of a sudden they'd collapse later, made the material really dangerous to use in people. But in 1949, Amberson went back and in the very first volume of the Journal of Applied Physiology described 7 patients who were treated with cell-free hemoglobin. And this is the critical one on the right. A woman postpartum who was in shock. They gave her plasma, they gave her glucose, they gave her blood in small volumes with no response. They gave her cell-free hemoglobin, and her systolic blood pressure went from 80 to 160, her diastolic from 50 to 110. She woke up, she talked to them, and 4 days later she died. Amberson said this shows both the potential and the risks of cell-free hemoglobin. A decade and a half later, Leland Clark published this picture of a rat floating in perfluorocarbon emulsion. To make this work, you have to have this under about 500 torr of oxygen to saturate the perfluorocarbon and to carry enough oxygen. And the perfluorocarbon has a viscosity of about 2. So the mouse is, but it's alive. And it can stay alive in that state for almost an hour. And then you reach in and take the mouse out, squeeze it a couple of times. It'll cough up a lot of the stuff and survive. You can't give this intravenously because, of course, it's a thick oil and it will have like fat emboli. But Geyer emulsified it in phospholipids and detergents and exchanged transfused rats down to, again, a hematocrit less than 1 and showed that under high oxygen tension, they could survive for 72 hours. The problem is that the perfluorocarbons he was using, perfluorodecaline and perfluorotripropylamine, literally persist in the reticuloendothelial system forever. And so once you've given an animal, the equivalent of about 40 grams of material, in a human, it's truly in your lymph nodes as a storage problem. Okay, but Fluisol was ultimately licensed by the FDA because of the recognition that when you expand the balloon doing coronary arterial balloon expansion of clots, if you perfuse this material through the catheter, you can prevent the ST elevations. Now, the cardiologist very rapidly figured out that, you could keep the ST elevations didn't matter if you kept the balloon up for less than 4 minutes. And so the material didn't get a lot of use. It was used in a randomized clinical trial in Jehovah's Witnesses. And the ultimate answer is that people with a hemoglobin of above 3.5 didn't seem to need it, and those below 3.5 didn't seem to benefit by it. And so ultimately the company took it off the market in 1989. Later, Alliance Pharmaceutical, a private company in San Diego, looked at all the potential perfluorocarbons that were available and looked for one that had the highest oxygen carrying capacity or highest oxygen saturation capacity, and also had a low vapor pressure, so that, you know, if you gave it, it would slowly vaporize and you would blow it out through the lungs. And so it didn't live in your body forever. The problem was they then emulsified it in egg yolk phospholecithin, which they thought would be the best possible immunologic wrapper for it. And by the time they gave more than about 3 grams of the material, it began to activate complement. And so the second graph on the bottom is the stock price going from $100 to pennies over a 2-year period. So normally you've got about 15 grams of hemoglobin. It carries 20 ml of oxygen, and that's normal. You can make hemoglobins, polymerize them, and we'll talk about that. You can, at lower concentrations of red cells, you carry less oxygen. And the plasma will actually carry a certain amount of oxygen just dissolved in water. Turns out you could not make the perfluorocarbons safely carry more oxygen than you carried in your own plasma. And so they died. Which left hemoglobin. Hemoglobin, as you know, is a tetrameric molecule, 2 alpha and 2 beta chains, and it has a the so-called beta-beta cleft down the middle. 200 molecules of water fit into that. And as hemoglobin goes through about a 15-degree shift, as you oxygenate and deoxygenate, those 200 molecules of water are pumped in and pumped out, and sucked back in. They're, you know, the molecule literally looks like it breathes. I'm going to use this cartoon as a stand-in for hemoglobin. You know, it's a globular protein, molecular weight of about 69,000, so it's the same size as albumin. And there's 250 million of them in each red cell. So each red cell carries 1 billion molecules of oxygen. And remember that red cells are about 65% of all the cells in your body. You know, you've got 25 billion red cells, 25 trillion red cells, and about 42 trillion cells in your whole body. This is the price we pay, you know, for breathing oxygen, for being able to do all the things that we do. And it represents the end result of 600 million years of evolution. As a molecule that started as a nitrate reductase has gone on to become an oxygen carrier. Hemoglobin, the tetrameric form, is only lightly held together, about a kilocalorie and a half of binding energy as van der Waals forces keeps it together. If you take it outside the red cell, where the mean spacing between 2 hemoglobins is about 1.3 diameters, it comes apart. And so, and when it comes apart, it exposes that interdimer face, which will bind to haptoglobin with the highest affinity of any 2 molecules known, suggesting that Mother Nature doesn't like free hemoglobin. Okay. In humans, there's a molecule called 2,3-DPG, which sits in the beta-beta cleft, and it pushes the oxygen equilibrium curve at the base to the right, reducing the binding of hemoglobin. But the real problem is that the binding of hemoglobin to oxygen is not perfect. You know, there are there's 250 million hemoglobin atoms inside each cell, and they're constantly binding and releasing the oxygen. I mean, that's what the equilibrium constant means. And so every once in a while, the oxygen takes an electron with it, and you make superoxide and methemoglobin. Now, the red cell is full of superoxide dismutase and methemoglobin reductase, and so it's constantly putting this process back together. But it takes energy. And if you try and store blood, you know, as you consume the glucose to provide that energy, you become acidotic. And the acidosis feeds back on molecules like phosphofructokinase, and you stop making as much energy, and then these toxic molecules persist longer. And as you stop getting rid of superoxide, you'll actually oxidize further from ferric hemoglobin, ferrous hemoglobin to ferric, and ferric to ferril, and you'll make hydroxyl ion, which is a powerful DNA and lipid destroyer. And that's, you know, ultimately why red cells have evolved to essentially have no internal organs and no DNA, you know, is to avoid the attack here. Okay, and hemoglobin not only binds oxygen, it binds other diatomic molecules, cyanide, carbon monoxide, and nitric oxide. And again, there's the rub. Nitric oxide is the endothelium-derived relaxing factor, and free hemoglobin will bind it. Okay, it's possible to modify hemoglobin. You can bind it together internally in the molecule, you can polymerize it, you can conjugate it. Molecules that will internally bind it include things like NF-PLP, 2-nor-2,4-meal-pyridoxyl 5-phosphate, you know, which will then flip the phosphate into the beta-beta cleft and not only cross-link the molecule, but also shift its oxygen binding somewhat. DBBF, which I spent 5 years of my life working with, is a dibromoaspirin that looks a lot like clofibrate, the anti-cholesterol agent, that will fit up again inside and can bind. And having a divalent molecule will literally cross-link the lysines at alpha 99 when you do it in the deoxy state. Somatogen, a company in Boulder, Colorado, was able to express hemoglobin in a polymeric form simply by putting an extra glycine in. And then again, it will make a molecule that is cross-linked, but is capable of flopping around. The alpha hemoglobin, which I say the Army developed with Baxter. Baxter took it to a human trial in 1999, managed to enroll 100 patients, 98 of them got treated, and the excess mortality was threefold. You know, and Baxter ultimately got out of the business, writing off $599 million and firing 600 people. For literally a decade and a half after that, I got phone calls from some of them in the middle of the night to tell me that I had done the right thing, that I'd ruined, you know, the great chance to save the world, and all of that kind of thing. it is possible to polymerize hemoglobin. And here they're using glutaraldehyde, a simple tissue fixative, but it's a bivalent aldehyde. And it will form shift bases with the lysines. And there are 50 lysines on the surface of hemoglobin. And so it randomly cross-links hemoglobins both internally and externally. And you can do this with bovine hemoglobin. You can do it with human hemoglobin and put a pyridoxal phosphate on it to, again, provide the effect of 2,3-DPG. And so these were products that were taken to human trials. It's possible to do this with essentially any aldehyde, like ring-open sugars. And the Canadian company, Hemosol and Hemolink, did this for a decade. And it's possible to conjugate hemoglobin. You can conjugate hemoglobin to polymers. You can conjugate polymers to hemoglobin. And the Sangart MP4 product was that one on the bottom. Okay, and again, Somatogen talked about making a continuously polymerized hemoglobin. This is what earthworm hemoglobin looks like naturally. And they were planning to make this product and humanize it to reduce antigenicity. Grow it in earthworms and actually try and make a blood substitute out of it. Okay, so in the late 1990s, these three products were poised to go into human trials. The polyhemoglobins from BioPure in Northfield and the PEG hemoglobin from Sangart. Now the problem with modifying hemoglobin is that much of that cooperativity is very rapidly lost. And so the binding curve changes from being sigmoidal to being hyperbolic. And so you lose oxygen carrying capacity at the top and the bottom. And if the problem with the single molecule was that the small molecules were highly vasoactive, then you had to get them out. And the only way to make the polymerized hemoglobins of relatively uniform size was to do size exclusion chromatography. Now the problem there is that became incredibly expensive. And you can treat every pituitary dwarf with about 1/4 pound of growth hormone, whereas 1/4 pound of hemoglobin wouldn't be a single person's dose. And so you've got to do huge volumes of this as a large volume parenteral. And then other problems, like bacterial contamination and endotoxin contamination become huge issues. But to get on with our studies, we actually built a sterile hemoglobin production facility, first on the Presidio of San Francisco, and then again in the Gillette building in Rockville, when we closed the Presidio under base realignment and closure. We would take 80 units of outdated human blood, collect them in a cold water-jacketed tank, add distilled water in the classic way. As the hemoglobin was released, we would be circulating it through a 500 kilodalton filter, which you can just see the edge of. And, and then you'd take it through a couple of purification steps, add the cross-linker, and ultimately collect it into liter blood bags for study. And we got to the point where we could get to about a 60% recovery. on this process. And at the same time, we learned how to make it more pure. Most of what you're seeing there on the left-hand side is the cross-linking material binding to other lysines. And so you get a decorated molecule as well as the cross-link. And we were able to get that down. And we were able to use these time-of-flight mass spec aspects. They were just being developed in Building 50 in the NIH, and we could go down there and run them, and being able to modify our process. Okay, so we made about 200 liters of sterile hemoglobin, for investigational purposes, and then we put it into animals and tried to discover toxicities. And of course we discovered that the nitric oxide binding caused the hypotension and vasoconstriction. And literally as we were doing this, this was being elucidated. Alvarez at UCLA won the Nobel Prize for that. The hemoglobin, as it falls apart, releases the heme, the iron, and these are profound free radical agents which especially damage neurons and endothelial cells. Leads to macrophage activation, to neurotoxicity, enhancement of endotoxic effects. And so let me kind of run through how we looked at all these processes and what we did. We made a model in swine, took about 20 kilogram young swine, would take out their spleen, put in catheters, and a week later we would take them awake to the, to an operating facility, and we would watch their blood pressure and other variables, measure blood samples at specific times. And what we would do then is after 2 baseline measures, we would remove blood from the animal, taking a total of about 40% of its blood volume, 10 ml over the first, per kilogram over the first 15 minutes, then 7, then 5, then 3. And as you can see, when you do that, the mean aortic blood pressure falls. And when you resuscitate with either 3 volumes of Ringer's lactate, the standard 3-to-1 crystalloid, 1 volume of albumin, 1 volume of hemoglobin in Ringer's acetate, and 1 volume of cross-linked hemoglobin in Ringer's acetate, what you would see is with the crystalloid and the albumin, blood pressure would return to normal. But when you use the hemoglobin, it returned 40 to 60 torr above normal. And so this is the hypertension that occurs. And when you measure that in the pulmonary artery, you get the same effect. But this time it's about 2.5 times as high. When you use the Swan-Ganz catheter and measure the cardiac output, what you discover is that as you resuscitate with the saline or the albumin, the cardiac output goes way up to compensate for the missing hemoglobin. Whereas when you put the hemoglobin back, the cardiac output does not increase. When you then, do the multiplicans of those and measure and calculate the systemic vascular resistance, what you see is the systemic vascular resistance has gone up by about 2.5-fold. And when you measure the pulmonary vascular resistance, it's up about 3-fold. And one of the animals that creates that disturbance there died in acute right heart failure during the course of the experiment. Okay, we looked at the dilution of the hematocrit as we did this experiment, and you can see that all the lines are essentially parallel, meaning that the resuscitations were approximately balanced. You can also see that when you put the saline in, it very rapidly third spaces, and you get volume contraction, but with a rise in hemoglobin. And when you put the uncross-linked hemoglobin in, it very rapidly dissociates and diffuses out of the vascular system. And so by 3 hours, it's largely been partially diluted. But the critical problem is that when you actually measure the oxygen delivery, we've been adding an oxygen carrier, but we do not increase oxygen delivery. The increase in the work and the decrease in cardiac output offsets the additional oxygen carriage. And so this means that you correct the shock at the same level. Water works as well as a hemoglobin solution in fixing shock. Okay, but for the free hemoglobin, the uncross-linked hemoglobin, which comes apart, as long as it's present in the circulation, it's damaging the kidney, and you can see an increase in the creatinine. And when you measured the output, the urine output, this turned out to be a high output renal failure. But as long as the hemoglobin is in the circulation, it's causing oxidative damage. And so when you measure creatine kinase activity, you're seeing cell breakdown and the release of this material into the circulation. And the LDH does the same thing. Okay. So normally your red cells are circulating, your endothelial cells are secreting nitric oxide, which goes to the smooth muscle cells and supports endothelium-derived relaxation. But as you increase the space in which the hemoglobin is occupied, you increase the clearance rate, the clearance volume in which nitric oxide is broken down. And so some of the paths by which nitric oxide gets from the endothelial cell to the smooth muscle cell, which may go into the circulation and back out, are lost. But then very quickly the hemoglobin will also leave the endothelial space and actually come in between those two and shut it off. And this can occur literally in seconds. Now, Alvarez won the Nobel Prize for nitric oxide by using rat aortic rings. And he'd have these tensiometers when it could show that if you took the aortic ring and used a wire brush and scraped the endothelial cells off, the tensiometer would contract. But if you took another aortic ring and perfused the de-endothelialized ring with the downflow from the endothelialized ring, it would relax. That's how we got the concept of endothelium-derived relaxation. And so using those techniques, we realized that the hemoglobins, when active, although when you block them with cyanomethemoglobin, which is very stable, wouldn't, this was active all the way down to nanomolar concentrations. And hemoglobin, at 15 grams per deciliter, 150 grams per liter, is present in millimolar concentrations. And so the toxicity is present at nanomolar concentrations, and the required physiologic concentration is at millimolar concentrations. This is a problem. And so when the Northfield Polyheme group came to the US Army and asked us to pay for their clinical trial, we did the simple experiment of just simply giving it to a series of rats who were 40% shocked. And it turned out that, you know, the negative controls, a bunch of other materials, the HBOC turned out to be the very worst thing you could try and give to the rats to resuscitate them. Now, you know, this kind of demonstration, people say, well, that's just in rats. We've seen it, you know, do other things. Okay. This is a skin window on the back of a Syrian hamster. You know, and there are the blood vessels that you can see. And when you do a fluorescein angiogram of them, you know, most of the capillaries line up in parallel with the muscle strands on underneath the skin. And you can then take another porphyrin, palladium mesoporphyrin, and flash it with light. It will dissociate the oxygen that it contains, and the rate at which the fluorescence decays is a measure of the oxygen pressure. And so, you know, here you have a quantum measure of oxygen content. And when you do this, what you can see is when you give, perfuse them with blood, the intermediate concentrations in the tissue are higher than when you perfuse it with cell-free hemoglobin. And when you do this in a ordered way, you know, starting with a hemoglobin of 13.9, the arterial, venous, and tissue concentrations of oxygen are what you would expect. And even when you drop it down to a hemoglobin of about 5.4, they stay at that rate. You are delivering enough oxygen to meet the tissue requirement. But when you drop the hemoglobin below the critical point, you know, the venous and tissue concentrations go away. And if you then reperfuse with hemoglobin, it does not get better. These are Choi cultures. A Choi culture, you take a fetal mouse brain and dissolve it in collagenase, and then plate it. And you'll get the neurons to settle on a plate of microglia. And then these two pictures, the one on the right was incubated in albumin, and the one on the left was incubated in hemoglobin. And you can literally see the places where tigrolysis, you know, occurred, where the neurons excited by the hemoglobin, which is an excitatory neurotoxin. You know, we put needles in them and watched, you know, these neurons simply have epileptic fits until they died. And you can do the same thing by disrupting the blood-brain barrier on one side by injecting hypertonic sugar into one carotid artery and then injecting cell-free hemoglobin. And you'll get neuron death on one side of the brain. These are neutrophils on the surface of endothelial cells in a paper by Steve McFall published, you know, out of our lab in 1994. And what he did was he took human white blood cells and separated them into the macrophages. And he would incubate the macrophages with human serum albumin or the alfalfa, the uncross-linked hemoglobin. And then he would wash those macrophages off and incubate them longer and then collect the supernatant and put the supernatant on the endothelial cell cultures for a while with or without anti-tumor necrosis factor. And then add the neutrophils and count how many stuck. And so what you see is that the albumin doesn't activate the neutrophils. The hemoglobin does, and it can be partially blocked with anti-TNF. And the same thing can be done with the chemokine interleukin-8. And so when you inject hemoglobin into rabbits, about 20% of them will develop this kind of severe vasculitis and get strokes, pulmonary emboli, and other consequences. Now, as I mentioned, haptoglobin binds the interdimer face of hemoglobin very strongly. And of course, when you cross-link the hemoglobin in such a way that you can't expose the interdimer face, you can't clear the hemoglobin through normal pathways. And so the same thing is true with the hemopexin, and you end up with these materials circulating longer, dumping their products in ways that can't be cleared normally. Bacterial endotoxin, has a phosphate on one end and a lipid on the other. If you mix it with hemoglobin, the phosphate sticks, normally it exists in my cells, but if you mix it with hemoglobin, the phosphate will stick into the phosphate binding site and present the lipid A in a much more exposed way. So these are rabbits, and if you give them bacterial endotoxin, really nothing happens. If you give them hemoglobin, they'll raise their blood pressure. But if you give them the two together, they become profoundly hypotensive and die. So if you think that the toxicity problem is bad, the pharmacology isn't much better. You've got something that lasts a short period of time that disrupts the relationship between intravascular volume and blood pressure and breaks down to cause toxic materials, the iron, the heme. But there's some beta chain breakdown products, beta hemopressin, beta hemorphin, that have independent pharmacologic activity. And the colloid osmotic pressure sucks water in, dilutes clotting factors, and just, designed as a resuscitation fluid, it actually interferes with resuscitation. Now, Hiromi Kasai up in Japan pointed out that, the larger you make the complexes of hemoglobin, the less high blood pressure reactivity there is. And so, we got into this issue of having to filter the hemoglobin to try and get it down into mixtures that had a molecular weight on average of about 250,000 Daltons, about 4 hemoglobin molecules. But if there are 50 lysines on the surface of every hemoglobin, then there's, 6,000 or 4,000 different ways to put 2 hemoglobins together. 16 million ways to put 4 of them together. And so you're really not dealing with a compound. You're dealing with a mixture of different agglutamers. Okay. Now, where does this leave us? The requirement for oxygen transport is absolute. So even a seriously toxic product may actually find some uses. And one can sort of play with it and say, okay, there are certain hemolytic anemias, where red cells can't survive. Maybe this stuff would be useful to carry oxygen to bridge these people over. There are situations in which you say, hold it, this doesn't make any sense at all because it only has a very short half-life. You'd give huge amounts of iron to buy life for a fairly short time. And so the situations where you want this material to work have to be very specific. Now, but others were vocal about the fact that these should work. And certainly, Paul Ness, the editor of Transfusion, Jeff Carson, who was writing the best stuff at the time, we're basically saying, look, this stuff is important, we need it. And Gene Moore, the editor of the Journal of Trauma, basically said the future is now. And so Northfield went to a human clinical trial. The other companies had managed to disable themselves. And the FDA basically said, look, if you can merely show equivalence, with Ringer's lactate, we'll give you a license. But 40 extra people died in the study. Okay. So Chuck Natanson at the NIH finally, he's the chief of critical care, just said, look, this is crazy. We have done 11 human trials with these products. There have been excess mortality in every one. If we did one more trial, it wouldn't change the meta-analysis. Stop. And these are the forest plots out of his work and all the various studies that were done, And so, people just said, okay, don't do this. But then, the companies went out of business. Northfield, after that human trial, declared bankruptcy and wrote off $227 million. But that left BioPure, whose trial was shut down by the FDA and ultimately said that none of the data was accurate, still persisted as a company and was willing to give away its product to people who wanted to use it. And good people, Jonathan Waters, who's again a co-editor with me on Transfusion, superb obstetric anesthesiologist at Pittsburgh, Jed Gorlin, who was president of the America's Blood Centers, and basically said, look, we've treated Jehovah's Witnesses with this material, and we've saved lives in people who would have died. Now, it's possible to wrap hemoglobin in liposomes. And this summer, Science magazine, published a There Will Be Blood article saying that these liposome-encapsulated hemoglobins, had properties that regular red cells didn't. They had long shelf lives and they were compatible because, they didn't have ABO types on their surface. But durable, does that mean it circulates as long as blood? No. And compatible, does it activate complement as many liposomes do? Unstated. And 15 years ago, I and Omo Efeabugo, basically pointed out that if you wrap hemoglobin in liposomes, it oxidizes twice as fast. So you can make these things. Okay, so where are we? Well, one, we're on the downslope of the scientific curve. Two, we have a material that is highly toxic but works in some people. And, I've watched a couple of people die of this material with acute pulmonary hypertension. I'm fairly negative on it. But it exists and it's there. Questions? Yes.
Thanks, John. That was fantastic. I don't think you covered it. I imagine it's something that has come up. We can give people nitric oxide. If that's getting consumed, can we give them enough, or is it just too toxic in the amount you need to give, or what is can you address that problem with inhaled nitric oxide?
Well, one, you're giving nanomoles of nitric oxide when you've got millimoles of hemoglobin. And, you know, it's, yeah, and its binding rates are extremely fast. You know, they're in the millisecond range. Yeah.
So I think it is clear, you very well established the toxicity of these compounds. So what do you think is in the horizon for transfusion, for this kind of idea of being able to provide blood to people that cannot receive blood for compatibility issues or for other things?
Well, the problem is the only material that's available is the biopure material, which, you know, the company gives away, but over the last couple of years, nobody's been able to get it. So, you know, there's this call that, you know, somebody needs to make this. And clearly the FDA is tired of dealing with biopure. You know, they lie, they falsify data, they, you know, are threaten people. They're, you know, they're not a good company to work with. Yeah.
And what are your thoughts about red blood cell production in vitro? That you can genetically modify, for instance, to alter the antigen expression, and then you can grow them like an artificial bone marrow?
I mean, you know, people are playing with that, and you know, for years I've been getting phone calls from, you know, the various groups around the world that do it and say, when we have this, what should we put it in? You know, and you know, the problem is that for the last 20 years, you know, you can collect a whole set of stem cells at a apheresis setting, and using all of the best modern techniques, make 3 ml of red cells. You know, so you lose more than that just collecting the stem cells. And, you know, we've been stuck for 20 years. I don't particularly follow the science, although, you know, I tend to be friends with all 3 groups that do it. You know, nobody is in a rush to, you know, say they have a product at this point. Yes?
As I recall, people have also looked at like fluorocarbons or others, like non-hemoglobin.
Yeah, and as I dealt with them sort of halfway through the lecture. You know, both the old perfluorooctyl bromine, or the new perfluorooctyl bromine, the old perfluorodecalan products. You know, the problem is one, they don't carry a lot of oxygen, so you've got to have a PO2 of about 500 to carry enough oxygen to make it work. The old molecules end up stuck in the reticular endothelial system for life. The new ones, you know, are slowly blown off, and then they become part of the ozone layer where they're, you know, fluorinated hydrocarbons. Yes?
John, thanks for the talk. I had probably a pretty naive question about sort of red blood cell storage. You know, you'd mentioned that sort of glycolytic activity leads to this negative feedback loop, which shuts down protection of red cells. That's supplementing with just ATP. I mean,
Well, ATP breaks down very rapidly in plasma. I mean, you know, your red cells actually secrete ATP as another mechanism, you know, that's part of endothelium-derived relaxation. You know, and ATP, you know, is secreted out of red cells. It doesn't get in very readily. And, you know, if you take red blood cells and stored them the way you do cell culture, you know, every 4 days you spun them down and replaced it, you can store red cells for 9 months with excellent recovery. You know, the fact that red cells store badly is because we store them badly, not because they're programmed to die. You know, it's now almost 40 years that I've been doing this. You You know, it's, you know, I wrote an article on it, you know, but, you know, there's thousands of articles out there. Almost all of them say this is a good thing to do because, you know, the companies put huge pressure on journals not to publish negative articles. And so, you know, this is the other side of the story. Yes, Hamilton.
John, you mentioned the last thing, which was the liposome coded hemoglobin. Do you think that possibly those trials might have a different outcome if they ever went to human trials?
Well, you know, I think all of these things can work in some people. You know, even in the trauma resuscitation trials, 85% of the people who got the product survived. You know, so that carefully chosen situations, and you know, like the you know, the patients with autoimmune hemolytic anemia who've been described, who've, you know, gotten through the acute crisis, you know, and people publish this stuff and say, see, this stuff actually allowed us to do something. And this is true. But you've then got to have somebody who's willing to manufacture large amounts of this stuff for a very rare use. And certainly it costs me about $4,000 a liter, you know, taking advantage of all of the freedoms that the Army gave me. I didn't pay rent, I didn't, you know, I got my blood for free. You know, this stuff would be expensive to manufacture, and it's a large volume pharmaceutical. You know, it's got to be done under exquisitely sterile conditions. You know, it's going to be, you know, if you made it in any useful quantity, it would be incredibly expensive. Thank you.